Hello, everyone. Today, we are going to learn and explore about DNA forensics and its significance in criminal investigation. Before moving forward, let's understand the word forensics. The word forensics derived from the Latin word forensis, which means of or before the forum. From identifying criminals to exonerating the innocent, from solving decades old cases to reuniting families, DNA forensics is just more than just science. It is a voice of evidence that never fades. So, in this presentation, we will explore the fascinating world of DNA forensics, its process, and significance in criminal investigation. So, let's dive into the microscopic world where every cell holds a story and every DNA strand is a key to unlocking mysteries. Historical background. Alec Jeffreys, also known as the father of DNA fingerprinting. Sir Alec Jeffreys is a British genetist known for inventing DNA fingerprinting in 1984. His discovery revolutionized forensic science, paternity testing, and biological research. DNA fingerprinting technique is a technique used by a forensic expert to identify an individual on the basis of their unique DNA profile. DNA fingerprinting was first used to solve a double murder case in England, proving a suspect's innocence and identifying the real perpetrator. Alec Jeffrey's discovery transformed forensic investigations and remains the foundation of modern DNA forensics. His legacy continues to influence crime solving, law enforcement, and genetic research worldwide. Now we will explore one of the most revolutionary tools in forensic science, that is DNA. From crime scenes to courtrooms, DNA has changed the way we solve mysteries and deliver justice. Introduction to DNA. DNA stands for deoxyribonucleic acid, is the blueprint of life. It carries the genetic instructions that make each individual unique. Found in almost every cell of our body, DNA remains unchanged throughout life, making it a powerful tool for identification. It stores the genetic information that transfer from one generation to the other. When DNA is found in the nucleus, then it is termed as nuclear DNA, and when DNA is found in the mitochondria, then it is termed as mitochondrial DNA. The double helical structure of DNA was given by Watson and Crick in 1953. Introduction to DNA forensics. What do you mean by the term DNA forensics? DNA forensics is the combination of biology, genetics, and criminal investigation. We can define DNA forensics as genetic analysis application in criminal investigation. In simple terms, DNA forensics is the use of DNA, the unique genetic code found in every living thing, to solve crimes and identify people. Why is it important to study about DNA? Because it helps to identify individuals based on their unique DNA profiles. DNA has several unique properties. Uniqueness. Every individual, except identical twins, has a unique DNA sequence, making it a reliable tool for forensic identification. Common sources. We can easily extract DNA from blood, saliva, hair, skin, bones, and bodily fluids. DNA testing process. So let's take a closer look at the step by step process of forensic DNA analysis. The first step includes sample collection. The first step is collecting DNA evidence from crime scenes, suspects, or victims. This can include blood, saliva, hair roots, skin cells, or even fingerprints containing touch DNA. Make sure proper handling and storage are crucial to prevent contamination. Next step is DNA extraction. In this step, DNA is isolated from biological samples. Next step is quantitation, where DNA is measured to determine its quantity and quality. This step ensures that there is enough DNA for further analysis and that the sample is not too degraded. Moving forward to the other step, that is DNA amplification. In this step, DNA is copied multiple times using a technique called polymerase chain reaction. Separation, where DNA fragments are separated by size using electrophoresis. Final step include analysis and interpretation, where DNA profiles are generated and compared to the samples. Example to understand DNA forensics. DNA forensics helps solve crimes by identifying people based on their unique genetic code. Let's understand how this works with a real life example. Imagine a burglary case where the suspect left behind a few traces of evidence, a half empty water bottle, and a blood stain on broken glass at the crime scene. A suspect is a person who is under investigation and is believed to have committed a crime. So, in this example, step 1 includes collection of DNA evidence, where investigators collect the water bottle and blood sample from the crime scene. They also take DNA samples from potential suspect for comparison. Second step includes DNA extraction and profiling. In this step, scientists extract DNA from the saliva on the bottle and blood stain. They perform short tandem repeat analysis, which creates a unique DNA profiles for comparison. Step 3 include matching the DNA. The DNA profile from the crime scene is compared to the suspect's DNA and entries in the national DNA database like CODIS. If a match is found, it links the suspect to the crime scene. Step 4 includes legal proceedings. This example shows how a tiny trace of DNA, invisible to the naked eye, can help solve crimes and bring justice. DNA forensics is a game changer in modern investigations, making sure no crime goes unsolved. Significance in criminal investigation. Why DNA forensics is so important to study in the field of criminal investigation. As we have already discussed earlier, DNA has their own properties. So, in this slide, we are going to study about significance in criminal investigation, suspect identification, and linking to crime scenes. One of the biggest advantages of DNA forensics is its ability to accurately identify suspects. A tiny drop of blood, a strand of hair, or even a fingerprint with touch DNA can link a criminal to the crime scene. DNA found in blood, hair, saliva, semen, or skin cells at a crime scene can be matched to a suspect. Exoneration of the innocent. DNA evidence is not just about catching criminals, it also protects the innocent. DNA evidence has helped overturn wrongful convictions through organizations like the Innocence Project. Wrongful conviction occurs when an innocent person is found guilty of a crime they did not commit. Victim identification. DNA forensics is also crucial in identifying victims of crimes, natural disasters, essential in mass disasters, terrorism cases, and war crimes for identifying human remains. When traditional identification methods fail, DNA can provide closure to families by confirming a person's identity. Sexual assault and paternity cases. In rape cases, DNA evidence can conclusively link the perpetrator to the crime, strengthening the legal system. Unlike eyewitness testimony, which can be flawed, DNA evidence is scientific and unbiased. DNA evidence is highly reliable and widely accepted in courts, establishing criminal patterns and repeat offenders. It helps in criminal profiling and behavior analysis of the criminals. So, in conclusion, DNA forensics has transformed criminal investigations providing unmatched accuracy in identifying criminals, exonerating the innocent, and solving even decade old cases. It serves as a powerful tool in modern law enforcement, ensuring justice through scientific evidence. Thank you for your attention. I hope this presentation has given you a deeper understanding of the role and significance of DNA forensics in the pursuit of justice. Thank you and bye bye.